Hello everyone, my name is Jorge Acosta and I'm going to talk about the Mendelian randomization study of ACLY and cardiovascular disease. Definition: ATP-citrullinase is an enzyme in the cholesterol biosynthesis pathway. Collect on the 3-hydroxy 3-methylglycerol coenzyme A reductase HMGCR. Inhibition of ATP citrullinase therefore reduce the plasma low density lipoprotein LDL cholesterol level by interfering with the same pathway as inhibition of HMGCR with a study. So ATP citrullinase is an emerging target for pharmacotherapy. It's unclear whether lowering LDL cholesterol level by inhibiting ATP citrullinase will reduce the risk of cardiovascular events to the same extent as inhibiting HMGCR with a statin. I'm going to show you two videos. So first, I need to talk about the Mendelian randomizations. The Mendelian randomizations, represent a novel epidemiology study design that incorporates genetic information into traditional epidemiology methods. A study based on Mendelian randomization will likely become increasingly common as genetic knowledge of health and disease expands with data from genome-wide associations, a study and genome sequences. Mendelian randomizations provide an approach to addressing questions of causality without many of the typical bias of the incompatibility of traditional epidemiology approaches. Okay, I'm gonna show you the two videos about the Mendelian randomizations. This is the first.
Epidemiologists are interested in understanding factors related to health and disease. The smokers tend to die younger than non-smokers. It certainly looks that way. But disentangling cause and effect can be difficult. Smokers are different on average from non-smokers. They're more likely to drink heavily and have less healthy diets. But even if we measure these confounding factors, we may not measure them perfectly, or there may be others we haven't measured. Also, as people become ill, they may cut down and give up smoking, which could wrongly suggest that reduced smoking leads to worse health. You could randomly assign 50,000 people to smoke and 50,000 people to not smoke and follow them up to monitor their health. This removes the possibility that any other factor could be responsible for any differences we see in their long-term health. But this is neither ethical nor practical. Fortunately, we've all been recruited into an experiment without knowing it at the point at which we were conceived. Our genes, which are passed on randomly from generation to generation, influence how much we eat, sleep, drink, smoke, and more. These genetic influences are not affected by anything else you may or may not choose to do in your life. They're not related to confounding factors. We can use this knowledge to learn about cause and effect, grouping people according to their genetic code. This method is called Mendelian randomization. For example, smokers who carry one version of a gene called CHRNA5 tend to smoke less heavily than those who carry a different version. When we group people according to which version of this gene they have, we find that the people with the version of the gene associated with heavier smoking will indeed die younger. But is the gene influencing how long we live in some other way? We don't think so. When we look at the same gene in non-smokers, there's no effect on life expectancy. The effect must be driven by smoking. Using this method, we can show that smoking causes lung cancer, heart and respiratory disease, and many other diseases, but does not seem to influence depression or anxiety. Mendelian randomization has already begun to tell us about factors that influence our risk of disease. Now we're using the same approach in other ways, to look at several risk factors together, and to look at what influences disease progression, which may help us to develop new treatments.
The second video is this.
The only randomization is using people's variation in genetics to approximate an exposure dietary exposure, for example. So the importance of Mendelian randomization is that it gives us the assessment of a dietary exposure that's independent of other dietary exposure. So it allows us to pinpoint a causal effect to a specific nutrient and not to other factors that are correlated with that nutrient in the diet. A nutrient that's important for cancer is vitamin D. And vitamin D we make partly from the sun, and we get it also in foods and some people from supplements. So there have been many studies associating vitamin D level with a lower risk of various cancers, particularly colorectal cancer. Now, because people, for example, people who exercise might have higher levels of vitamin D because they're more exposed to the light or to the sun. We are not sure that the vitamin D itself is protected, or it may just be an indicator the person exercises a lot or has a good diet. Now, we can use Mendelian randomization by looking at genetic variants that help determine vitamin D levels. So some people just for genetic reasons have higher vitamin D levels than other people. And if we can show that those people are at lower risk for cancer, that would give us more direct evidence that vitamin D is indeed directly related to cancer, and not just because it's a marker of a good diet or people exercising, factors like that.
Nutrition is ubiquitous in what we do, and first of all, nutrition is very important for cancer research. So we have several links that nutrition is associated with cancer, but at the same time, it's not easy to study nutrition and diet in general, because most of the studies we design, we ask the participants to report what they have been doing in the near past about their food intakes, which is not easy, and obviously it can create misreporting, so people may not remember what in general they eat over the last year.
We've learned a lot from traditional methods of studying nutrition and cancer, and we know very well dietary patterns that are likely to be protective for cancer or increased risk of cancer if they're bad dietary patterns. But when we get to specific nutrients, there's a lot of controversy, and a lot of studies have not confirmed what had been initial leads. Like, for example, we thought initially that vitamin E might be protective for cancer, but further studies show that it did not. And part of the problem was that when people, for example, have high levels of vitamin E or vitamin C, or people that might have better diets. So it's been really hard to tease out the specific benefit of vitamin C or vitamin E independently of just being a marker of a good diet. So Mendelian randomization gives us potential to more specifically look at the specific components of whether it's vitamin C that's related to the lower risk of cancer. The traditional methods have many advantages for looking at the overall dietary patterns, but Mendelian randomization gives us another tool to look at more specific factors.
Application in medicine, ACLY inhibitor reduced LDL cholesterol with similar efficacy have HMG CR inhibitor studying with a comparable overall risk profile. The pros and cons. Pros ACLY variant associated with decreased LDL cholesterol level are associated with a decreased risk of cardiovascular events, provides genetic validation for ATP citrate at a therapy target. So the cons, genetic variants that mimics the effect of therapy again cannot anticipate potential drug-specific effects of a therapy that is not related do its mechanical actions. I'm gonna show you two more videos.
In epidemiology, Mendelian randomization is a method of using measured variation in genes of known function to examine the causal effect of a modifiable exposure on disease in non-experimental studies. The design was first described by Gray and Wheatley, 1991, as a method for obtaining unbiased estimates of the effects of a putative causal variable without conducting a traditional randomized trial. These authors also coined the term Mendelian randomization. The design has a powerful control for reverse causation and confounding which otherwise bedevil epidemiological studies. An important focus of observational epidemiology is the identification of modifiable causes of common diseases that are of public health interest. In order to have firm evidence that a recommended public health intervention will have the desired beneficial effect, the observed association between the particular risk factor and disease must imply that the risk factor actually causes the disease. Well-known successes include the identified causal links between smoking and lung cancer, and between blood pressure and stroke. However, there have also been notable failures when identified exposures were later shown by randomized controlled trials, RCTs, to be non-causal. For instance, it has now been shown that hormone replacement therapy will not prevent cardiovascular disease, as was previously thought, and may have other adverse health effects, Rizzo et al. 2002. The reason for such spurious findings in observational epidemiology is most likely to be confounding by social, behavioral, or physiological factors which are difficult to control for and particularly difficult to measure accurately. Moreover, many findings cannot be replicated by RCTs for ethical reasons. Mendelian randomization is a method that allows one to test for, or in certain cases to estimate, a causal effect from observational data in the presence of confounding factors. It uses common genetic polymorphisms with well-understood effects on exposure patterns, for example, propensity to drink alcohol, or effects that mimic those produced by modifiable exposures, for example, raised blood cholesterol, Caton 1986. Importantly, the genotype must only affect the disease status indirectly via its effect on the exposure of interest. Because genotypes are assigned randomly when passed from parents to offspring during meiosis, if we assume that choice of mate is not associated with genotype, panmixia, then the population genotype distribution should be unrelated to the confounders that typically plague observational epidemiology studies. In this regard, Mendelian randomization can be thought of as a natural, randomized controlled trial. Because the polymorphism is the instrument, Mendelian randomization is dependent on genetic association studies having provided good candidate genes for response to risk exposure. From a statistical perspective, MR is an application of the technique of instrumental variables, Thomas and Convey 2004, Ideles and Shi in 2007, with genotype acting as an instrument for the exposure of interest. MR is based on a number of assumptions. These include that there is no direct relationship between the instrument and the dependent variable, and that there are no direct paths between the instrument and any potential confounders. In addition to direct effects of the instrument on the disease misleading the analyst, misleading conclusions may also arise in the presence of linkage disequilibrium with unmeasured directly causal variance, genetic heterogeneity, pleiotropy, often detected as a genetic correlation, or population stratification, Davy Smith and Ibrahim 2003.
This is the other video.
Hi, I'm Perry Wilson, and this is Impact Factor, your weekly commentary on the most interesting or important articles appearing in the medical literature. This week, how Mendelian randomization works and what it can tell us about vitamin D, milk drinking, and bone fractures as we check out this study appearing in the BMJ. So, what is Mendelian randomization? Broadly, it's a statistical framework that attempts to figure out if some exposure causes some outcome. Causation is the holy grail of medical research because, for example, if low vitamin D levels cause bone fractures, then treating low vitamin D levels would prevent bone fractures. So let's say you want to know whether low vitamin D levels cause bone fractures. You measure vitamin D levels in a bunch of people, follow them over time, and see if those with lower levels develop more fractures. But be careful, what you just described was an association between low vitamin D levels and fracture risk. You know very well that other factors like diet, exercise, and sun exposure can affect vitamin D levels and might independently change your fracture risk. These other factors are known as confounders, variables that are linked with both the exposure of interest, vitamin D level, and outcome of interest, fracture. And they are the bane of observational research. We have a slew of statistical tools to try to account for confounders, but none are perfect. Now, if you really want to get at causation, you should do a randomized trial, but these can be tricky and very expensive. It's not always ethical or feasible to randomize people to certain exposures, which is probably why we have yet to see a definitive trial exploring the efficacy of absinthe use on painting ability. Mendelian randomization gets around these issues via an interesting epidemiologic trick. We know there are a host of things like lack of exercise that might be associated both with low vitamin D levels and fracture risk, but there are genes that are associated with low vitamin D levels too. Some people are born with gene variants that predispose them to lower vitamin D levels throughout their lives. Whether you get one of those genes or not, practically speaking, is random. It has nothing to do with eating habits or exercise. It's an accident of birth. So if low vitamin D levels cause fracture, then a group of people genetically cursed to be predisposed to low vitamin D levels should have higher fracture rates. On the other hand, if low vitamin D levels are merely a marker of other healthful behaviors, being born predisposed to have low levels will have no effect on fracture risk. This is the genius of Mendelian randomization. We use the genes people have had their whole lives, genes that never change, to get around the mess of the myriad life choices that affect measured vitamin D levels. The genius of the BMJ manuscript is that they used genome-wide association data from over 500,000 individuals to perform Mendelian randomization analysis, not just on vitamin D levels, but on a host of classic fracture risk factors, including dairy intake, diabetes, age at menopause, and more, all things that are associated with fracture, but have not been shown to cause fracture. This is a shotgun approach to find something we can change that will actually impact the fracture rate. What they found was that out of 14 risk factors for fracture, only one, bone mineral density, had strong evidence of causality. Grip strength, a proxy for muscle mass, had weak evidence of causality. Remember, they are not saying that these other things aren't associated with fracture. They are saying that changing the other things won't change the rate of fracture. So we get a ton of information from this study. It suggests that, for example, while those who have earlier menopause are more likely to have a fracture, delaying menopause is unlikely to make a difference in the fracture rate. What about vitamin D? Well, this study would suggest that all those vitamin D supplements we take aren't doing much to decrease the fracture rate, and that finding broadly aligns with randomized trials that have examined vitamin D supplementation. So that's Mendelian randomization. I hope I've brought a bit of sunlight to the issue. For Medscape.
Issues in Ecuador. The principal problem in our country is the poor research culture. Actually, we are not experts in Mendelian randomization studies, so we cannot make a study about the ACLY. I think some results that may help our public health. I'm going to show you the last video about the education and coronary heart disease Mendelian randomization study.
Is the leading cause of death in the world. Approximately 1 in 2 men and 1 in 3 women develop heart disease during their lifetime. In an effort to learn more about this disease, scientists notice that people who spend more time in education are less likely to develop heart disease. This finding is interesting. However, what so far has been a mystery is whether this relationship is due to cause and effect. For example, Other factors can give the impression of a cause and effect relationship, when in fact, these other factors are causing both heart disease and education. One way to check if more education prevents heart disease is to do a randomized controlled trial. For example, take a group of young individuals, some with high physical activity, some with low, some with good diets, some with bad. Randomly shuffle them into two groups. Give one half more education and give the other half less education. Then wait about 40 years to see which of the two groups develops more heart disease. Such a trial would give us the most reliable evidence on whether more time in education is protective of heart disease. However, this trial is virtually impossible to do because of the long wait time. So researchers of the current study did the next best thing. They mimicked a trial by looking at randomization that occurs in nature. In every human, all genetic material is randomly allocated from their parents, including 162 DNA markers linked to education. Some people randomly receive more of these 162 DNA markers, while others receive fewer. Just like in a randomized controlled trial, the beauty of this natural trial is that the allocation of educational DNA markers is not linked to any other factors, such as diet or physical activity. Researchers then asked the question, do people with more of these 162 DNA markers, and therefore slightly more education, develop less heart disease later in life? The answer is yes. The group with more education developed about 30% less heart disease. This finding suggests that longer education prevents the future development of heart disease. This means that doctors, public health experts, politicians, and civil society groups who advocate for more education may also be doing a big favor for the health of our hearts.
The conclusions, genetic variants dominate the effects of ATC strategy like inhibitory and steady appear to lower plasma. LDL cholesterol enabled by the same mechanism of actions and were associated with a similar effects on the risk of cardiovascular disease. For unit decrease in the LDL cholesterol level, therapeutic effort to reduce ATP, citrate activity reduce LDL